Welcome back. This is lecture 3, part 3, and I'm still Dr. Beal, and we're going to start looking at biological macromolecules. I've broken this lecture into 6 segments, and so this is part 3 of topic lecture 3. I did this because I think there's a lot of information, but also there's very easy ways where I want you to sort the information. It's a good way to break it out. Traditionally, this is one of the longest lectures of the semester. It actually takes a period and a half. So normally we would get halfway through this lecture and then you'd come back the next day. Give yourself a little bit of extra time to watch and think about these because it is sort of designed to be a little bit longer. Macromolecules, policy acknowledgement, do the policy acknowledgement. Macromolecules are really big molecules. We had a practice question at the end of the last topic. Video, I'm running out of things that call it videos. My topic is my big idea, and I group them into lectures. We've had this question about which were polar. I think you got it. I'll talk about it in my review session. I think you got this. But I would also be very interested in seeing what kind of organizers you've come up with for this. It's important to me that you figure out the best way to deal with the volume of information you're getting. Because as we go on, I'm going to stop helping you so much. I'm going to continue to give you information. And I want you to be comfortable with easy one-page organizations of these. If you found you took a bunch of notes and had to sort through them later, sort through them as you read them, give yourself a general idea. What were we talking about? What outcome did this part of the lecture address? And then see if you can answer that outcome within your notes. Make a page in your notes that says this is the answer to that outcome. This is how I compare and contrast functional molecules or functional groups. So this part of the lecture, I don't know, I think I might be over this. I think I move it over later. We're going to talk about how we build biological molecules. This is actually, we're still starting the ideas behind cell respiration. In cell respiration and photosynthesis, some of the things we do is we build molecules and we break them apart. They're biological molecules. They're things like carbs that you take in when you eat or proteins that you use to do things in your body. Macromolecules are big molecules, but they really mean within the context of biology. Macromolecules are built up of subunits, single building blocks. We call them monomers. Mono means one. Mer actually means piece. So these are one pieces. Those building blocks are then built together to make long chains of pieces, long chains of mers, polymers. Poly means many. I like to think about these polymers as being sort of like Legos. A Lego block is one piece. You have to hack it apart with a saw if you want to break it up. If I build my Legos together, I can build a lot of different complicated things depending on the order of assembly, how many pieces I put together. Maybe I can make a Lego house, or I can make a Lego Millennium Falcon. It depends on what order I build my mers together, my pieces. Biological molecules work that way as well. 3 of the biological molecules, we'll have 4, are polymers. They're made up of many individual monomers built together. Carbohydrates, things like sugars, proteins, things like enzymes, and nucleic acids, things like DNA, are built up of smaller building block units. Lipids are biological molecules, but they never form polymers. I don't have any monomers. I don't assemble monomers into long chains where their function changes depending on what order I put the monomers together in. That just doesn't happen. Lipids are always just hydrocarbons. Lipids are lipids. If I put a bunch of lipids together, I just get a blob of lipids. If I assemble a bunch of amino acids together, I can get lots of different proteins depending on how I assemble them. So here, we've seen this one. Any guesses? Has an amino side. Acid or base? A carboxyl side. Draw a box around it, acid or base. I assemble them around this center carbon. I have an amino acid. It's a good example of a monomer. Ooh, functional groups. I have amines and carboxyls. I have something that is going to change here. That's what R means. I got a carbon, a central carbon. We call it the alpha carbon, and it has a hydrogen on it. So let's take some hydrocarbons. Let's build one. We got some carbon with some hydrogen. That's a hydrocarbon. We'll not worry about this R group for now. I'm going to add a couple functional groups. I added an amine and a carboxyl. This thing together, this single unit is a monomer. It's an amino acid in this case. It's built of a carbon with some functional groups attached. I'm going to use these, lots and lots and lots of these, to build a larger molecule. I'm going to build a polymer. So how do I do that? Here I have one monomer. This is a one amino acid. We're not going to worry about which amino acid. I don't care. It's amino acid 1. And here's amino acid number 2. I want to join them together. I got to use some kind of chemical reaction, right? And I'm certainly going to have some kind of enzyme. And we'll talk about enzymes, but something's going to help me here. And I'm going to use the enzyme and a process called dehydration synthesis to fuse these two molecules together. Now, before we even go anywhere, synthesis means joining things together, right? If you take a course in English language and you read a lot and they ask you to produce a synthesis at the end, they want you to summarize and get the main points. Synthesis, when we talk about it in biology, is taking things and building them together. How about dehydration? What does dehydration mean? When you go to the shore and you're dehydrated, what have you run out of? Water, right? You've run out of water. So we're going to remove water to build these two molecules together. Here's the water. I drew this myself, can you tell? Here's the water. I'm going to remove oxygen and a hydrogen from monomer number 1. I'm going to remove a hydrogen from monomer number 2. And they're going to snap together. I do it real fast. Here I'll remove these. The water goes away. And now you can see that I've done this art myself. I've formed my two monomers into a polymer with water as a side is released. If I have this polymer and I want to break it back into two monomers, guess what I'm going to do with enzymes? I want to break it. Breaking, and I'm going to add this water back in because I want to put them back the way they used to be, right? I want to put them back into two amino acids. So I'm going to use a process called hydrolysis. It's hydrolysis when we put it all together. This is water. Lysis means break. I'm going to use water to break, with the help of enzymes, this molecule apart. So here I animated it for you. Yeah, I know it's like watching Disney, but better. The water goes in, and it breaks up into two molecules again. What I'll have you do if you're kind of sketching on your own, this is a great place if you have a whiteboard or you have something you can erase. Try this. Draw these molecules, remove the oxygen and the hydrogen from one, the hydrogen from the other, snap them together. You are dehydration synthesizing. Take that water back and put it back into the molecule, snap them in half, and you're doing hydrolysis. Hydrolysis is another name for digestion. Your mouth is full of water and enzymes. You put sugar in your mouth. One of the things that happens is hydrolysis. You use water to break up the molecules in your mouth, and you start to digest them. Hydrolysis takes polymers and it breaks them back into monomers. Dehydration synthesis takes monomers and it forms them back into polymers. You have an outcome that asks you to sequence polymerization, that's the building of polymers, sequence how dehydration synthesis works. And then a second outcome asking you to compare and contrast hydrolysis and dehydration synthesis. Come up with some categories to help you tell them apart. My best suggestion is to pick one of them to start with and practice it. Practice breaking it up or building it back together, whichever one you think is the best. And then learn the second one. You learn the comparison reaction. That'll help you keep it straight in your head. So this is a summary. This is not a great organizer. This is sort of a summary of everything we've talked about. Here's dehydration synthesis. I take 2 monomers, I remove water, and I make a polymer. I'm going to use a lot of enzymes. Those enzymes that help with dehydration synthesis are often called dehydrogenases. They allow for dehydration synthesis. If I have a polymer, I can add water and some enzymes, and I do hydrolysis, water breaking, and I can end up taking my polymer and converting it back into monomers. And enzymes are going to help me do this as well. These enzymes are called hydrolyzes. And you'll start to see a pattern here. This ase at the end of word, ase, is telling me that it's an enzyme. It's doing something to my molecules. That's it for this video. I want you to get comfortable. Those reactions are going to appear over and over again for the rest of the semester and next semester. Those are the basic ways that we build things and break things apart. And I'd like you to get them straight before we come back to look at the biological macromolecules. The next video is just going to be about carbohydrates. And I'll see you soon. Do your policy acknowledgement. All right. This is the last set of videos I will constantly irritate you this about, but I really want you to do it. And I'll see you soon. All right. Bye-bye.